This first lecture is actually on just a general introduction to metabolomics, and it's really to try and get people acquainted with the field. Some of you are probably more expert in NMR, others are perhaps more expert in metabolomics, and so this is just trying to, I guess, level the playing field so everyone knows a little bit. And I've just colored things here to indicate the first 4 or 5 things that we're going to be doing. And what's colored in green are the ones that we'll do today. And then things in red are tomorrow, and things in blue will be the last bits on Friday. So I'll begin with a picture that sort of depicts the way that we like to think about metabolomics and proteomics and genomics. This is really to indicate how things play out with respect to the connection between the different omes, the genome, which is sort of the base for the pyramid, and genes code for proteins. Proteins are really there to facilitate chemistry, to accelerate it, and the chemistry all happens with the metabolome or with metabolites. The genome, as most people understand, is critical for coding many of these things. The metabolome, in many respects, acts as the interface between the genome and the environment. And that's partly why I've shown this issue or picture of arrows going up the pyramid with increasingly intense color, to basically indicate that the environmental influence is much greater as you go up the pyramid. What you've been eating or drinking or breathing is profoundly affecting your metabolome. The fact that the metabolome can change with what we see or experience or eat or breathe makes it a particularly sensitive indicator of the environment. But it's also influenced by the genome. And so the fact that the metabolome is at the interface between the genome and the environment means it's a really useful indicator of phenotype and is commonly used for phenotyping. Metabolome is also used as a way of detecting what's happening at the genome. Metabolites amplify what's happening in the genome. Metabolites are sometimes called the canaries of the genome because a single base change at the genome level can lead to a 10,000-fold change at the metabolome level. The other thing to remember is that the metabolome is quite specific to different organs. So the brain has a very different metabolism than the liver. The liver has a different metabolism than the skin, and the skin has very different metabolism than the gut. However, the genomes for most tissues are identical. And so this means that our picture, our vision, our view of the world, particularly from a genomic perspective, is colored by thinking that all cells are the same, all tissues are the same, and all organs are the same. And that's obviously not true. Different organs produce different collections of metabolites, so they are bathed in different metabolites. So the kidneys produce urine, bone marrow produces blood cells. We've got the brain producing cerebral spinal fluid, saliva glands producing saliva. Each of those is essentially bathing specific organs and using those fluids for detection, and metabolite detection or analysis really gives you a detailed picture of what's happening with those organs. So physiology and environment are things that, in fact, profoundly affect the metabolome and for which metabolomics is very good at detecting. So I think in comparison or an analogy to what metabolomics is, you can use this definition that's commonly used for genomics. So it's a field of high-throughput science, high-throughput DNA sequencing, where you're aiming to identify and characterize all of the genes in a given cell or tissue or an organism. So all of those genes represent the genome. So for metabolomics, essentially it's the same thing. We just substitute a few words. We change the words from genes to metabolites or small molecules. And the same concept applies. And you could do the same thing with proteomics. So this is where the analogy is. But obviously, we're looking at different kinds of molecules. So in metabolomics, we're looking at small molecules. The formal definition is any organic and even inorganic molecule with a molecular weight less than 1,500 Daltons. This is kind of an arbitrary cutoff, but it actually is sufficient to include almost all lipids and things that are smaller. But it also ends up including certain small peptides of about 10 or 12 residues or so. It can include short oligonucleotides. But it also includes many of the things that we think of as metabolites in general. So that can be including sugars and organic acids and nucleosides and ketones and aldehydes. Those are the endogenous molecules that our bodies create, synthesize, or metabolize. But it also can include other things that we expose ourselves to, the foods we eat and the food additives, alkaloids that might be found in certain fruits or vegetables. There's obviously toxins and pollutants and drugs, over-the-counter and prescription, along with their drug metabolites. It also includes all of the compounds that we find in terms of the microbiota. And when I'm saying human, I can also include mammals, I can also include plants. These are all part of what many living systems have. They all seem to have a combination of endogenous, exogenous, and microbial products. These are the things that you can detect with metabolomics. And just because they may not be endogenous or coming from your own organs, it doesn't mean that they're not metabolites. They're often very important. There's also an issue of detectability. This is a little different than the genome. The genome is a very finite entity. It really doesn't change with respect to concentration, or doesn't change with the environment. With the metabolome, it's changing with the sensitivity of the instruments we use. And as instruments get more and more sensitive, we're discovering the metabolome is getting bigger and bigger. So right now, the lower limit of detection is around picomolar, but who knows where next year or 10 years from now we'll be at. And as we get down in the detection limit, certainly the metabolome will grow significantly. So the metabolome, different from metabolite, is that collection of small molecules. And as I said, it includes those endogenous and exogenous molecules. And ultimately, it's defined by our detection technology. For this course, we're going to be talking about NMR. In some regards, it's the least sensitive technology, so in that respect, it makes metabolomics a little simpler. But in all cases, the metabolome will always be ill-defined. It's not as firm or as finite as the genome. Now, there are a number of different kinds of metabolomes. When we think of mammals, the current estimate is there are perhaps about 60,000 known chemicals that are in humans and horses and dogs and rats and mice. So in that regard, mammals are relatively simple because if you compare to all microbes, the chemical diversity is probably greater. They occupy far more diverse environmental and ecological niches, and they're important for transforming or biotransforming many of the things on Earth. They're the things that made the Earth inhabitable. But what's perhaps most interesting of all is the diversity of plant metabolites, and that the number of known compounds that seem to derive or have been identified from plants is approaching 300,000. So why is it that plants have a much greater metabolome or more complicated metabolome than animals or microbes? This has to do with the fact that plants can't run away. So they have to wage chemical warfare to fight off predators or herbivores or infectious agents. And so they've had to create, over their evolution, thousands of secondary metabolites, which are used to protect the plants and to protect them from pests or other invaders. Microbes can swim, humans can run, and so that basically means we don't have to have very complicated chemistry. Now, if we look at the types of compounds, and this is, I think, relevant to all of us because we're all human, I hope, this is a measure of the type of compounds that we'll find in the human body, and it's probably somewhat similar to other mammals. We know, at least at current count, that we've identified formally, this is likely to expand, but there's about 20,000 endogenous metabolites that we've cataloged and they're located in a database called the Human Metabolome Database. And they range in concentration from low picomolar or high femtomolar up to almost molar levels. Urea is the most abundant metabolite in terms of concentration in the body and can get up to 100 millimolar. If you include water, you can get up to 55 molar. But the endogenous metabolites are the most numerous and span the greatest concentration ranges. When we started looking at the metabolome, we realized that, in fact, we weren't just measuring endogenous metabolites. Very soon we started seeing people with drugs, prescription and over-the-counter. And typically, drugs span a slightly lower range, typically at the micromolar level. And they're not as numerous as endogenous metabolites. There's about 1,500 or 1,600 drugs that we know of that are approved for use. And we've put a lot of that information into a resource called Drug Bank. Another thing that we also became aware of was that people eat food. And many of the foods that we eat are not obviously composed of endogenous chemicals. They're made of phytochemicals, salads, and fruits, and vegetables, and coffee, and tea, and many other things. And then we put additives to improve the taste or color or flavor. And the result is that there's probably on the order of 30,000 known chemicals that are in your food, on which you will eat today or yesterday. And again, they span a range very similar to the concentrations we find in drugs, from picomolar to micromolar typically. Drugs get metabolized, and they produce drug metabolites. We've also tracked those. That's kept again in the Drug Bank database. And then at the lowest end of the concentration curve are toxins and environmental chemicals, pollutants, and so on. They're hopefully at nanomolar levels and below. If they're higher than that, then you're in trouble. And we've tried to track a lot of those compounds through a database called the Toxic Exposome database, and that tracks about 3,600 of them. Now, these are the things that are known, and in fact, the number, as I say, is approaching more than 40,000, and soon will be above, I think, 50 or 60,000. But we also know that there's a lot more that we can't see, a lot more that we don't know. This is often called the dark matter of the metabolome, or the unknown unknowns. So we can take some measures of particularly the variety of acyl chains that can be found in lipids, and we can do some calculations, and we can estimate that the number of lipids that might be in humans are probably over 100,000. We also know that even though we've tracked maybe 1,200 drug metabolites that are registered or known, just the fact that there are 1,500 drugs and the assumption around 6 to 7 different metabolites for each drug tells us there should be at least 10,000 drug metabolites. We know that many foods are transformed, and so if there's 30,000 different chemicals in foods, those are also transformed, again, quite substantially. And there could be 10 to 15 different metabolites for each compound. So these are called secondary food metabolites, so we're up to 500,000. And then there's the compounds within our bodies that are transformed by our liver enzymes, by our microflora, and also by our own endogenous enzymes, which aren't perfect and will periodically transform things just because they want to. And in fact, that is what we call the metabolites of metabolites, and this may represent the largest component of that dark matter. So altogether, these things may sum up to something on the order of 1.5 to 2 million different compounds, none of which are in any database, none of which have been characterized to any degree. So this is truly the unknown, and this represents sort of the limit of detection that we have. In the case of instrumentation, it's usually in the nanomolar or subnanomolar range. So that's kind of a list or a tally of the different types of small molecules that we can see, or maybe not yet see, but can detect. It's also important to remember that small molecules, as numerous as they are also important. If we look at the standard common clinical assays that are used by a doctor's office, or if you've ever had a doctor's test run by, almost all of them are actually small molecule tests. True, there are protein tests and there are DNA tests, but the common ones, overwhelmingly, the ones that are billed to the health agencies, are typically small molecule tests. Around 90% of all drugs are small molecules. Certainly the ones that make the news are the big protein drugs, but it's still a case that most drug companies make their money, and most of the drugs that most people take are small molecules. What's more is that half of all the drugs, it's actually close to 55%, are actually derived from pre-existing molecules. They are inspired or represent slight derivatives of those pre-existing molecules. So many of our brilliant ideas for drug development actually come from nature. If we look at genetic diseases, we know that about a third of all genetic disorders actually involve diseases of small molecule metabolism. And then within our bodies, small molecules serve as not just the bricks and mortar of the cells, but they also serve as the cofactors and signaling molecules for thousands of proteins in thousands of pathways, most of which are not in any book or any online database. So small molecules are really important. As I said before, small molecules are also the canaries of the genome, reporting what's happening with the genome, and historically have been used by clinicians for more than 100 years to help measure the needle in the haystack, looking for or identifying those single point mutations that cause very profound defects or disorders or diseases. As a field, metabolomics is very time sensitive. If you eat something, your metabolome changes, and if we could track all of the compounds that are going in your body and changing, being transformed, you'd see things climbing and falling and going up and down. If you did the same thing and were tracking your proteome, you might see maybe a boost in insulin and a slight perturbation in sort of a few gastric peptides. If we tried to track your genome after a meal, nothing would or should happen. So the temporal sensitivity of metabolomics is both an advantage and sometimes a disadvantage. If you're looking for something that's stable and you want to characterize, genomics is great. It doesn't change over your lifetime. If you're wanting to track things that are happening over minutes, seconds, or hours, metabolomics is often one of the best ways to do that. So that temporal sensitivity is an important thing, and often people get confused or confounded by that, and it's important to remember. In terms of metabolism, catabolism, and anabolism, some of you have probably seen pictures or charts like these hanging in walls. In many respects, metabolism is understood. Many of the critical features of basic metabolism were worked out in the 1950s and 60s. So in that regard, we can connect many metabolites to many genes and proteins. We know rates, and we know the cofactors, we know transformation, constants, everything else. It's exquisitely detailed. And the information that we have on metabolism is far greater, far more detailed than we have for just about any other field of biology. Because we know metabolism so well, we also can use it to help understand the other omes, the proteome and the genome. And in many cases, people doing metabolomics end up connecting these three together in ways that help bring together this concept of systems biology. So instead of thinking of the world divided as genomics and proteomics and metabolomics, through bioinformatics and through much of the work pioneered by people in metabolomics, we're starting to blend them all together and creating the field of systems biology. In many respects, most systems biology or most successful system biology models grew out of metabolic modeling. The metabolic reconstructions that have been done over the last 5 or 10 years are perhaps the most sophisticated examples of systems biology. And again, they were driven by metabolomics, but used and incorporated many principles of proteomics and genomics. There are obviously lots of applications in metabolomics. We'll visit or revisit some of these over the next few days. One of the most obvious and frequent and most widespread application is genetic disease testing, newborn screening, actually. Metabolomics is used in nutrition, food analysis. It's technically used in any kind of blood analysis or urine analysis or even cholesterol testing, and we'll talk about those. Drug companies use it to measure and assess drug compliance. Metabolomics is also used in MRI or MRS and chemical shift imaging. It's used to help in transplant monitoring. It's used in clinical trials, phase 1, 2, and 3. In the beverage industry, people will use it for both assessing and testing and fermentation monitoring. In the analysis of nutraceuticals, these are often critically used where they depend on metabolomics. Water quality testing, water assessment, water purity assessment is an active area, particularly in Europe. And there's petroomics or petronomics, looking at a composition of chemicals in oils, which is also very complex. So that's a brief background on metabolomics. We'll get more information over the next few days. Now we're going to sort of dive into the methods associated with metabolomics, because it's not a one-size-fits-all. This is a typical workflow for metabolomics, where it involves taking samples. It could be plant, animal, microbial. If they're solid, we'll extract them. If it's possible, we'll try and get fluids from them. In the case of animals, it's often very easy to get things like urine or blood. In all cases, what we're trying to do, basically, whether it's a solid or through the extraction or through biofluid collecting, is we're trying to get fluids, because chemistry is more easily done in fluids. And largely what we're doing in metabolomics is analytical chemistry. And the analytical chemistry we do is basically based on liquid chromatography, gas chromatography, mass spectrometry, and NMR spectroscopy. So this route of starting with a sample, extracting, and going down to some analysis is something that's been going on for decades. So in that regard, there's nothing new to metabolomics. On the other hand, the central advance that has allowed metabolomics to happen is in this last step. And we're going to focus a fair bit on this course and over the next few days about those steps, which allows you to take a mixture spectrum of complex organic compounds, and identify and quantify those many components. Because historically, what analytical chemistry was all about was separating so you could work with pure compounds. And it was only when you had everything very simple that you try and identify. But in metabolomics, we're working with the complex mixture straight off, and we're not even trying to necessarily separate things. And in NMR, we never separate things. So this is, I think, the great advance that's happened, and it's needed a lot of software, a lot of databases, a lot of innovation. And it's still happening, so it's not fully mature. But it's this step, as I say, which has been the critical advance that's allowed metabolomics to sort of move from the back room to the front room. Now, even with the efforts of many thousands of scientists, metabolomics is still very limited in its coverage. So it's now quite routine to sequence entire genomes, human genomes, rat genomes, almost any mammalian genome, for between $1,000 and $2,000. And we can typically measure not only the billion or 3 billion bases, but we can identify all 22,000 genes. In proteomics, standard techniques typically allow people to measure between 5,000 and 10,000 proteins quite routinely. In metabolomics, you're considered to be doing very well if you can identify and quantify 200 compounds. So in terms of the completeness of coverage, metabolomics still falls far behind what other omics techniques can do. And the reason has to do a lot with chemistry. In genomics, people only have to deal with basically 4 kinds of bases, and they're very, very similar chemicals. In proteomics, you have to only deal with 20 amino acids. Yes, they're in different combinations, but the chemistry of amino acids is well known. In metabolomics, we're dealing with hundreds of thousands of different chemicals, and something on the order of 10,000 different chemical species, which means that they have very, very different behaviors, very different features. So the complexity, the chemical diversity in metabolomics is what makes it so much more difficult, and is what is fundamentally limiting our coverage, at least for now. So in genomics, you can basically use a single instrument, a DNA sequencer, and you can sequence all you want. With proteomics, you can use one or two types of instruments. In metabolomics, you have to use a whole range. You have to use a whole range of different chromatographies, capillary electrophoresis, liquid chromatography, microfluidics. Use a whole range of different types of mass spectrometers, some for measuring polar molecules, some for measuring metals. Of course, we use NMR spectroscopy, sometimes to characterize the actual structure of compounds. We have to turn to crystallography. We, of course, use GCMS. Many groups also use infrared spectroscopy. A whole arsenal of equipment that's very diverse and typically would fill many rooms and which would often extend beyond most capabilities of most analytical labs. But all of these can be and are used. So I'm going to talk about these in a little more detail. I'm not going to talk a lot about NMR. That'll be the next lecture. But to dive in some of the other technologies for metabolomics, just so you have context and can kind of appreciate what's typically used in a full-bore metabolomic analysis. So the first one we'll talk about is chromatography, which in essence involves the separation of components and mixtures. So in chromatography, you're dealing with what we call a mobile phase, passing through a stationary phase. So the mobile phase could be liquid, it could be a gas. And really what you're doing is you're getting differential partitioning between the mobile and stationary phases. The stationary phase may have features that could be charge, or could be porosity, hydrophobicity. And the mobile phase interacts with the stationary phase based on those physical features. So within chromatography, people can, even in the world of metabolomics, use both column and thin layer chromatography. It could be either liquid or gas. Affinity chromatography, ion exchange, size exclusion. Most people use reverse phase, but you can also use normal phase. Or Hillick. There's also high pressure or just gravity to move the mobile phase. The most common technique still in metabolomics is high pressure or high performance liquid chromatography. So that was developed in the 1970s. It's a very mature technology. And instead of using gravity to push things down, as most of us might use for protein purification, you use high pressures. And you use very, very small, very pressure-stabilized particles. And with this approach, you can not only separate compounds, but with the appropriate detector, you can detect at very, very low levels. And depending on the matrix you use, you can separate both polar and nonpolar compounds quite effectively. So with HPLC, 3 modes that are typically used. Reverse phase for separating the nonpolar molecules. And so typically you're working with a nonpolar stationary phase, things that are decorated with sort of greasy compounds, and a relatively mobile, often organic, polar mobile phase of methanol and acetonitrile, sometimes water thrown in. Normal phase is rarely used, but it is used for separating nonpolar molecules. Actually, I should have said, but you use, instead of a nonpolar stationary phase, you use a polar stationary phase. Hillick is picking up popularity. It's allowing people to look at a lot of the things that would come out in the void volume of HPLC. And this is something that many people forget, is that most molecules in the body are very polar. Most metabolites are very polar. And so if you want to look at most molecules in the body, you should probably be using Hillick of some kind. So it's ideal for separating polar molecules, and it's working with both a polar stationary phase and a relatively polar mobile phase. So depending on the geometry and the types of particles in your column, you can get very different kinds of efficiencies. So short columns don't generate great separation. Longer columns, which take longer to run, give you excellent separation. But you can also get benefits if you shrink the size of the particles. So a typical size of maybe 5 micron particles, the separation isn't great. If you can shrink the particles down to 1 or 2 microns, you can get great separation on short columns in short times. And this is the principle behind ultra performance, or UPLC, ultra pressure, because the small molecules require, or small particles require higher pressures. So UPLC, an extension of HPLC, basically is the same as HPLC, but higher pressures, smaller particle sizes. Standard HPLC process involves mixing a solvent with your mixture, putting it through a pump, adding your sample, and pushing it through your HPLC column, and associating it with the detector. The detector can be a mass spectrometer, a fluorimeter, a UV spec, even an NMR. And through that, you generate a chromatogram with peaks basically corresponding to one or more compounds. So HPLC is central to MS. So when we say LCMS, it's really HPLCMS or UPLCMS. And in that regard, mass spec still requires basically working with relatively pure components of a mixture. Now, you can do liquid chromatography. You can also do gas chromatography. And this is sort of the forgotten cousin in metabolomics. It's the older form of chromatography. And in some respects, it is more developed and more consistent. So in this case, to get gas chromatography to work, you have to vaporize your sample and turn it into a gas. And instead of using an inert liquid, you use an inert gas to push things through. So it could be helium or sometimes nitrogen. So the column itself is a hollow tube. It's not filled with small particles. The tube is lined with essentially polymers that can be hydrophobic or sometimes hydrophilic, and it's the gas phase molecules that interact with the sides of that tube that allows you to get that partitioning. So instead of columns that are maybe 10 or 20 centimeters in length, columns in gas chromatography are up to 10 meters in length. And instead of having diameters of a few, 1 to 2 centimeters, they have diameters that are measured in millimeters. So they're very, very thin, long tubes. Now, many compounds are not easily turned into gases. Volatiles, things that have, boil at room temperature, those are good examples. But it's also possible to volatilize things that don't turn easily into gases by derivatizing them or chemically reacting them with trimethylsilane or other derivatives with silanes in them. And that can convert an amino acid, an alkaloid, a fat into something that actually is volatile. So key to much of what's done with gas chromatography is this process of derivatization. You're reacting, in this case, this is an example of a sugar, where you'll, first of all, modify some of the sugars with methanolysis, and then volatilize with this trimethylsilane derivative that produces this compound, which is a derivative of the clean sugar, but now has many TMS derivatives. Sometimes it may have 1, 2, or 3, or combinations of those. So this is what allows you to measure many metabolites via gas chromatography, but also makes gas chromatography complicated because you have to do chemistry, and chemistry is never at 100%, and it also means that any given compound can have 1, 2, 3, or 4, or 5 different varieties, depending on the level of derivatization. So this is just an indication of what you're typically dealing with. Gas chromatography, you'll have a sample, a mixture, and you're pushing it with helium instead of with acetonitrile or waters as with liquid chromatography. It goes through this column or tube, and things are partitioned based on their interaction with the sides of this column. What you get is a very detailed, very sharp, highly resolved chromatogram. And this is the amazing thing about gas chromatography, is that the resolution you get through it is always better than what you get for liquid chromatography. The plate count is many times higher, and it's far more reproducible than liquid chromatography. So those are some real pluses for it. As I said, the columns that you use are really not columns, they're tubes. So the polymer coatings that are going inside, a polysiloxane, and typically, rather than running at the full length of the room, they coil the tubes up so that they can fit into relatively compact instruments. So fundamentally different columns, and the columns can be derivatized with different compounds for different applications. Some are best for lipids, some are best for gas phase volatiles, others for other applications. So whether it's liquid chromatography or gas chromatography, ultimately you have to detect what comes out. Otherwise, they're just sort of pushing stuff into the drain or into the atmosphere. So what we do is we attach them to mass spectrometers. So this is, in fact, the most common method for doing metabolomics, attaching it to a mass spectrometer. And mass spectrometry, at least relative to NMR spectroscopy, is simple because all you're trying to do is weigh things. So a typical mass spectrometer is a size of maybe a table that you'll see. Some of them can be vertical, some of them can be horizontal. This is a time-of-flight mass spectrometer. They're usually configured with computers, and they have various electronic systems or magnetic systems to help with the measurement of molecular weights. So the whole point of mass spectrometry is to not only detect but also identify compounds by their mass. So, I don't know, there's 30-some people in this room. All of you probably have, at least if we measured it down to the gram, a different weight. And so if we had your name and your weight written beside it in the registration sheet, we could just simply weigh you and blindfold ourselves and identify who's who. And the same thing is obviously true with small molecules. Small molecules have different molecular weights. So whether it's ethanol with a molecular weight of 46.1 or L-DOPA with a molecular weight of 197.2, they're identifiable by their molecular weights. So with the best instruments in mass spectrometry, we can measure their molecular weights, or technically their mass to charge, or M/Z values, to within 1 ppm, or 0.0001%. And that is actually accurate enough to determine a molecular formula, C6H12O6. We can get that from the mass alone. For big molecules like proteins, typically it means you're determining molecular weights to within a Dalton for larger proteins. So Dalton is essentially the equivalent of 1 proton or 1 atomic mass unit. So you can attach a mass spectrometer to a gas chromatography system. That's called GCMS. You can attach a mass spectrometer to a HPLC or UPLC system, and that's called LCMS. You can also attach 2 mass spectrometers together, and that's called MSMS, and that's so-called tandem mass spectrometry. And so in gas chromatography, we separate the volatiles, we identify by their mass. Liquid chromatography, we attach them to HPLC, we identify by mass. In MSMS, we separate fragments of whole molecules and identify them by their fragment masses. And this is an important part of mass spectrometry. And it's actually a very important part of gas chromatography mass spectrometry. Most chromatography mass spec involve, or even GCMS and MALDI, involve sending something that gets ionized. So an electrospray ionization, it's sent through a nozzle with a strong voltage applied across the ends. It goes through a mass analyzer, which might consist of a magnet or a set of quadrupoles or an ion trap, again, some kind of a electrically controlled device, which separates things based on their mass to charge ratio, and then they fly against a detector. Unfortunately, mass detection is incredibly sensitive. So a mass spectrometer alone can just be used as a detector for what's coming off your chromatogram or chromatographic equipment. But it gives you the bonus that it also tells you what the mass is of that peak. So a mass spectrum, if it's done through electron ionization, where there's fragmentation that's happening because electrons are colliding with the molecule, or a mass spectrum taken from a triple quad mass spec, will produce this sort of thing. Very, very sharp peaks, much sharper than what you see with HPLC or GC. And these peaks are not measured according to time, they're measured to mass to charge. And this is the parent ion molecular weight of aspirin. And then what you're seeing are peaks corresponding to different fragments of that aspirin. And aspirin breaks up in a predictable way. And that predictable fragmentation pattern allows you to not only identify it not just by its parent ion mass, but by the fragment pattern. Now, this is a low-resolution mass spec. You can get to higher resolution mass spec. And you can measure down to as I say, 1 ppm. So higher resolution mass specs are called time of flight instruments. Lower resolution instruments are typically quadrupole or ion trap instruments, where they can measure down to a Dalton as opposed to 0.001 or 0.1 or 0.01 Daltons. So a mass spectrometer, as I say, has both ionization source, an analyzer, and a detector. There's usually the separation, chromatography at the beginning, and then computers at the end, and always lots of vacuum pumps. And those are the things that usually break down. Mass spectrometry is an inherently dirty system, and that you're putting stuff in, burning it up. Eventually, after a while, the sources or the analyzer get dirty as well, and you have to clean them. So the ion source is critical, and many of the different configurations we have for mass spectrometry have to do with the name of the ion source. MALDI, ESI, API, EI, CI, those are all the different ionization methods that are possible. So electron ionization is a hard ionization technique. It's a matter of impacting the molecule with high energy, 70 electron volt electrons that fragment and shatter the molecule. They give you structural information. Chemical ionization is a less common method, but it's not as hard in terms of what's being derivatized. It allows you to often see the parent ion mass. Spectra are a little simpler, sometimes a little more predictable. Then there's two soft ionization techniques which were critical for the development of proteomics, but have also been widely used in metabolomics: electrospray ionization, or ESI, and MALDI. And as I say, these are primarily used for proteomics, but they are increasingly and frequently used for metabolomics, and they give you spectra of the parent ion molecule and some of its adducts. So if you're using electron impact or electron ionization, if you start with something like methanol, it will produce these types of fragments. First, an ionized form, which flies through the quadrupoles, but also fragment in kind of weird ways, where you'll form double bonds or triple bonds, or hydroxyl groups are flying off. And these are detectable, and they produce very characteristic peaks that tell you that the molecule is there. I've mentioned electrospray ionization before. It typically we feed through something through a nozzle. It's almost like an aerosol nozzle with an electric field applied. It sprays things off. Another approach to soft ionization is to put stuff on a matrix, an organic derivative. Cyanohydroxycinnamonic acid is often used. Polyphenols can also be used. And shine a strong UV laser, and it causes things to blow up. And the things that blow up are ionized, and they can now be sent through your mass analyzer. So the mass analyzers also vary, and depending on the type of electronics, the resolution that you want, and how much money you want to pay, you can use a whole bunch of things, from time of flight to ion traps to quadrupoles to magnetic sectors. The original mass spectrometers were made up of big, strong magnets. They're still used today, typically for isotope analysis, so magnetic sector analyzers. Very, very high resolution. Quadrupoles, these are typically attached to GCMS instruments, or very cheap and very old LCMS instruments. So they have relatively low resolution. You can put several quadrupoles together, and you get a triple quad. And this is the most popular mass spectrometer in the world, actually, and used widely in drugs and in newborn screening. Time of flight historically was the most accurate, but then was replaced by things like Orbitraps and FTICRs. These are expensive instruments, so generally the higher resolution, the more money. And with those, you can get near exact mass, remarkable precision, and they're remarkably robust instruments as well. So that's a quick run-through of mass spectrometry. I think, obviously, we could spend weeks talking about it, but this is just a quick update to make you aware of what's possible. I'll talk more about NMR in terms of its use. Most of you, I think everyone here, has probably played a little bit on an NMR spectrometer, but the technology is obviously very different. Samples are worked in liquid. We don't do separations. We have big magnets. We're basically using radio frequency pulses to be able to detect and measure the resonance signals. And Like everything, then we use computers to analyze the output. So the three technologies I've mentioned, NMR, GCMS, and LCMS, are different in principle, but also different in their sensitivity and the type and numbers of metabolites they can measure. The most sensitive of all is LCMS, where it's not unusual for people to get down to nanomolar or even picomolar concentrations, and where people can routinely see 5,000 or 10,000 features. In GCMS, it's not unusual to get down to sub-micromolar or hundreds of nanomolar in terms of concentration, and to detect and sometimes identify and quantify up to 200 or 300 compounds. In NMR, the typical sensitivity is about 5 micromolar, and the number of compounds that people can typically identify and quantify is between 50 and 100. So NMR is the least sensitive, LCMS the most sensitive. But with increasing sensitivity means the fact that you're starting to identify things that are in the grass, things that we don't know. The proportion of compounds that we can truly identify in LCMS is about 5%. So while we can see 10,000 features, we're usually happy if we identify 50 compounds. On the other hand, with NMR, we might see 10,000 peaks, and we can basically assign every peak to a compound. So the proportion which you should or could be able to identify is about 100% in most samples. In that regard, NMR allows you to deal with the known compounds. It allows you to not only identify, but to quantify and to work with a lot of known biochemistry. The unfortunate thing with LCMS is that you're dealing with things that can take sometimes years to identify, and it's sometimes very difficult to publish results from LCMS metabolomics. Intermediate is GCMS, and where you get, again, about half the compounds or signals you see are identifiable, the other half are not. Now, it's not to say that you shouldn't do NMR or you shouldn't do LCMS. In fact, all three technologies provide complementary pictures of the metabolome. And in our own lab, we use all three techniques routinely. So it is critical, I think, to identify that all three have complementary approaches, all three are useful. These are just some slides that sort of highlight what you typically see with NMR. NMR focuses more so on water-soluble compounds and is really great at identifying sugars and organic acids. GCMS, you can get both hydrophobic and water-soluble compounds. LCMS, or GIMS, is actually really great for more hydrophobic or lipid-like molecules. There are different types of samples that you can use. Some are better for different systems. There are different limits in terms of the sample volume. LCMS or DIMS, very small sample volumes, whereas NMR, 100 microliters to 500 microliters is typically required. GCMS, more intermediate. In terms of sample preparation time, run time, data analysis, they're all different. The limits of detection are all different. The number of metabolites you might see and identify and quantify also varies. But even though they vary considerably, the number of compounds that you'll identify between each system is tiny. So again, this just emphasizes the complementarity. Each is giving you a different picture. What's more is that even with the overlap, it helps you cross-check. And this is something that many people don't appreciate. By using two or more methods, you can verify the reproducibility, authenticity of the measurements you're making with the other technique. Now, what you can see from here is that many of these things take a long time. That it's not seconds, it's not even minutes, it's hours for each sample to be analyzed and to characterize them. And even for NMR or for LCMS, they still take a fair bit of time each. Automation is typically greater for LCMS thanks to the huge efforts going into proteomics, where automation has been driving a lot of activities. But we'll talk about some of these new technologies later on about how this is also enhancing runtime and automation elsewhere. So if you do NMR-based metabolomics really well, you can identify between 50 and 200 metabolites, and you can quantify them as well as identify them. If you do GCMS really well, you can potentially identify up to 200 compounds, but in terms of quantifying them, it's typically a lower number, between 20 and 120. The different LCMS methods, in terms of identifying and quantifying things, it's somewhere between 150 and maybe 300. Lipidomics, it's possible to identify thousands. It's hard to quantify them. You generally group them into classes, so not individual compounds. But it's still very impressive. And this is, I think, where a lot of advances have happened and some very compelling results are emerging from lipidomic studies. And then typically, if you want to look at other types of compounds, some people will develop very specialized assays that are HPLC-based, GCMS-based, NMR-based. And they'll be for classes of 10, 20, or 30. So this is more typical of targeted metabolomics. Now, in that regard, there are two routes to metabolomics, and I'll mention these a few more times over the next few days. One is called the untargeted method, and the other is called targeted, but you can also distinguish them part by ones where chemometrics is heavily used, and others where deconvolution is heavily used. So in the chemometric methods, often you don't think about identifying things. You just treat this as a collection of peaks, and you distinguish between classes of this spectra and classes of another type of spectra and see how they cluster or group. And again, there'll be certain peaks that are different, but you may not ever know what those peaks are. In the quantitative, targeted deconvolution methods, you start with your spectra and immediately identify everything and quantify everything. And now you have lists of metabolites and their concentrations. And from that, and with those lists, you can actually do all kinds of things, which also include statistics, not unlike what's shown here. So the untargeted approach, as I say, involves generally collecting many, many samples into different groups, doing not only the collection, but a data reduction process to identify the more significant peaks, the more significant features. And then once you've identified the significant peaks, starting here with thousands, you may be down to 10. It's up to you to try and figure out what those peaks are. Sometimes you can do it, sometimes you can't. In the case of targeted metabolomics, and you can do targeted both with mass spec and with NMR, you actually start immediately off with the identification and quantification. You produce lists of metabolite lists. Then you do statistics. And then because you've sort of sped up a little bit, you can actually do a lot more biological interpretation because all of the compounds are known. And you can go and look through pathway databases and other literature and interpret things at much greater detail. So you can break down metabolomic analysis into sort of 4 steps, whether it's both targeted or untargeted. Typically, the first one with targeted or quantitative metabolomics, as I like to call it, is to go from spectra to lists of compounds. Now, it may be compounds that are identified, or it may be compound lists of features with their intensities. So if you don't know what it is, at least you can say it's a peak, and I know it's a peak, and it's at this elution time or this chemical shift, and this is its intensity. So it's still a feature, but whether it's lists of compounds and the concentrations, or lists of features and peaks with their intensities, you're still working with lists. From the lists, you can use statistics to shrink them down. So you may have hundreds of lists with hundreds of features or molecules. You want to shrink it down so it's a smaller list, perhaps 5 or 10, of significant features or significant metabolites. And we'll talk about how this is done, but this uses multivariate statistics. Once you've reduced your big pile of compounds or features or these massive lists to something that's a few, you can start doing analysis. You can get to look at the pathways, maybe metabolic or signaling pathways. And from the pathways, you can go a little further and you can start identifying biomarkers or models or systems biology models. And we'll briefly talk about some of those things, but that represents the interpretation part of metabolomics. So typically, where most of the time is spent is in this phase, going from spectra to lists. One that's most challenging for people if they don't know statistics, is going from lists to significant metabolites. And the most interesting and fun part are going from those significant metabolites to the pathways and the biomarkers. So those are the things that we're going to talk about over the coming few days. And with that, I'll wrap up here.